All right, welcome back to the Weekly Boost podcast, episode 46. Today, just Austin and I again, to your fortune or misfortune, I don't know, however you want to perceive it. But today, so we were talking about what was this in kind of the beginning of the summer, as we were like wrapping up our May campaign. It's like, well, like we've kind of been focused on like the history of this, the history of that, and it really like coincides with what like I'm doing. So to kind of flip things on its head, Austin's had the great idea of last week's show about going to Mars and STEM education. So if you haven't checked that one out, I would advise you do that, after this episode or whenever you have time. But, just to kind of give you an overview of what to expect today, we're going to talk a little bit about, this idea of a microbial world and how important microbes are to the Earth and in our lives. Kind of touch on what Austin's been preparing here to get published, really exciting stuff. And then kind of connect both of our fields, and you can see how some of the things are kind of coinciding with what we're doing and how everything's kind of interconnected. So if that interests you, stick around and we'll have a good discussion here. What do you think, Austin?
Yeah, I'm excited for it. Real quick before we jump into a lot of this stuff, one of the most difficult things that I've realized getting into the topics of this discussion is trying to maintain credibility and trying to maintain like the idea that, hey, we're actually some sort of, accredited individual studying this stuff. And, with our common nomenclature or vernacular or however we talk, we kind of maybe, not misguided truth, but sometimes we tend to forget things about like, the things that we're talking about. So just, again, take this for face value, really. Don't try to dig too deep into it. I may or may not be right or wrong with some of the things that I say, but it's the stuff that I've kind of taken away from, what I've been researching, as well as some of the stuff that I've been just looking at an educational level. So,
and I think that's a different episode that we should cover sometime soon, this idea of imposter syndrome and not like realizing that we're like credible in what we're doing. Like I go through that almost every day. It's
God. Yeah.
It's terrible. Yeah. And we'll save that discussion for a different episode. But, it is tough to try to, remember everything that you learned when it comes to microbes, and especially in today, like if you miscue once, it's like everybody comes at your throat. But
exactly.
I think that, like you said, the important thing as we kind of move forward in conversation and future episodes and past is just realizing that like we're here to learn. Everybody, we're curious about things, and we like talking through different challenges to life and just everything around us and talk through some things. And it's like we said, it's a grounding experience, and we hope that you guys can take something from this and appreciate the conversation. But
yeah, for sure. So kind of just get started. One of my most favorite topics to discussion about microbes or microbiology or whatever, the invisible world that we call microbiology, is the idea that when you think of like living organisms on Earth, you think of like plants and animals, right? But then you can get down into, some more different like avenues for life that exists on Earth. And a little fun tidbit that I've taken from my professor at St. Norbert is 15% of the world's biomass is actually bacterial. And so you never think about that. Like biomass is the weight of all living organisms that inhabit the Earth. And so you think of obviously trees and, like large mammals or just animals in general, they make up a good portion of it. But something that is invisible to the naked eye makes up 15% of the weight of the biomass. And it's like that it's an astronomically difficult number to comprehend, because again, it's something that's invisible to our eyesight. Like we can't look at these things without either staining them with like crystal violet or crystal violet or safranin or something like that, doing a gram stain. But also we need a really high powered microscope sometimes. And so again, availability to see these organisms, it's hard to believe that they actually are there. Let's just say that. The only time that you see them are in cases of infection. That's kind of when they rear their heads, is when there's an assay available that shows their physical presence on Earth. And so that was the first hurdle to get through, is that the idea of like, anywhere you go, there's millions upon billions of microbial cells on your body. For example, you're covered in them, in your digestive tract, it's loaded with them. So it just the understanding that they're everywhere kind of is the first step of this. And I thought that was something important to mention because, again, we kind of like the idea, I'm saying we as humans, we like the idea of seeing is believing, and things that are invisible to the eye are hard to believe in. I'm just going to say that right now. And so, yeah, my professor also really likes saying it's a microbial world. We're just living in it. It's kind of a, comical way to think about it, but it gets around the same point. So,
yeah, it's about 80% are plants, and then
yeah,
the next closest is 15 for bacteria. Just looking at this article, it's and then fungi and archaea, protists, and animals, and then viruses. And we mentioned that whether viruses are living or not, and here it is. It's classified as biomass. So,
yeah,
no, I'm interested. So, I'll try to shy away from like history stuff, right? But
yeah,
this, like, think back to like what you're saying about it's hard for you to like fathom something that you can't necessarily see.
yeah,
And bacteria to me is always like the forgotten about bio section, right? Like I took for zoology and
yeah.
things like that, and you just kind of like write it off and you kind of take it for granted. But it's I don't want to like try to interview here, but like talk a little bit about like why it's so important, the ways that we have like tailored our needs, like to use by like bacteria and microbes. Like how have like humans better suited it for ourselves,
Yeah.
just kind of throughout, I guess,
Yeah.
history. And
Well, I think the best way to go at your question is when you're working with bacteria, most of the time, like the stuff that I've done is you're actually working with a live organism. When you're going into, comparative zoology and like those classes where you have dissections, right?
Mhm.
Those are euthanized animals taken from a large facility that, allows for these animals to reproduce and create more of them. So they're already euthanized. They're deceased bodies that you're examining and understanding the anatomy and whatever and making connections to humans. Whereas with bacteria, you don't think about it as much, but when you're growing them up, these are living organisms. And it's one of the first experiences as a scientist when you're taking, your microbiology course as a, sophomore in college, or when you're taking like the brief general micro section in your high school biology class, like you're learning about something that you can, granted in a lab setting is when you can actually study these and analyze them safely. But it's one of the first instances where you're dealing with something that's living. And so there comes struggles with that, obviously, but there also comes sort of an appreciation because it is in fact something living, in that you become so infatuated with it, you realize that everything in your life has some sort of bacterial kind of history. Like you think, like I mentioned before in the beginning of the episode, like your gut, your digestive tract, how important bacteria are to that. Like we wouldn't know these things unless we studied them, and that directly relates to us as humans. And like same thing with the skin, and like using staphylococcus and streptococcus bacteria in your skin as defense mechanisms for foreign pathogens from entering, because they like the dry salty surface of your skin. Like we wouldn't know these things without being able to study them, and because we're able to study them, we can realize that, hey, these are living organisms. We can actually use them in a living way to understand how they grow, reproduce, not reproduce, but how they kind of form and create more cells, because there's different, they do it a little bit differently than we do as eukaryotes. But it's kind of a neat connection, because you think of like the stuff that we did, like you dissect a frog. Well, that frog has already been dead. It's like you can't really understand how it's living unless you go out and study it in the wild in a jungle somewhere. Like you have to go to these places, like bacteria everywhere. I could take, I could swab my ear, or I could swab like underneath my fingernail and grow a culture of something and analyze it, because we relate so closely to we relate and rely so closely on these organisms. So it's pretty neat.
Well, what I've kind of, when we were at Ripon, or when I was at Ripon, I don't think you were there yet, but when I'm learning about some of these things, and like gut microbiome transplants and
Yeah,
other things like that, like
those are fun.
that is like a really cool. I don't know when that, all kind of manifested and people understood that, but the uniqueness of gut biomes and the difference between people, and like you said, its importance, I think is something that not a lot of people either know about, but this idea that you can have, transplanted,
Yeah, yeah, I know
and
exactly
be
what you're
able
talking
to
about.
like introduce healthy bacteria
Yeah,
into your gut and your GI tract. And I think that's something that's
yeah,
especially promising for,
yeah,
and I don't know what word I'm trying to use, but like gratifying maybe for
yeah,
like researchers to be
yeah,
able to do something like this. And like you said, there's different treatments all that have to start.
yeah.
And like you said, cultivating bacteria is hard enough, like
It
on
is.
media, but
Yeah,
when you have to, like what you're doing when you're introducing certain things to fish and bacteria and seeing what happens, I think the fragility of maintaining these like these microbes in cooperation with some of these experiments is really remarkable.
well, and you mentioned, a human health relationship, like with the sort of transplantation of the human, gut or microbiome. For example, when an individual comes into the hospital with a C. diff infection, normally people try to treat it with antibiotics, and we'll get into that. I'm going to talk about that in a little bit,
Yeah.
but C. diff is an endospore forming bacteria, and so antibiotics can only do so much, especially if they're not targeted correctly, if they don't target the right, if they don't get the strain exactly right of C. diff. And so C. diff, it retains itself inside your gut even after the antibiotics cycle goes through. And so then these endospores still contain the materials for regeneration of bacterial cells. And so then C. diff comes back. And so that's one of the reasons for these, I'm just going to say stool sample transplants is actually what it is. Like, cutting out the malarkey. It's actually the scientific name for it. It's a fecal transplant. So that's how you can kind of regenerate somebody's microbiome in their gut, because depending on the specificity of antibiotics, when you ingest antibiotics like penicillin, depending on what kind of biome you have in your gut, it can wipe out the good bacteria as well. And so that's why you hear doctors saying, like, I know, Grant, I'm not a doctor, I'm going off of word of mouth from this, but like replenishing the healthy biome in your gut with like eating yogurt and stuff like that has the bacterium that help us, because believe it or not, bacteria help us a whole heck of a lot more than we encounter them as being harmful to us. Now, granted, the risks with the harmfulness, tetanus on rusty nails and like, eating dirt, like, or having dirt ingested in your body, like, yeah, that poses a risk. But at the same time, you're living, walking, breathing with your skin, and you have staph on your skin that doesn't infect you. And now MRSA is an up and coming thing where like 1/3 of the human population has it present on their skin just because of antibiotic resistance coming up from Staph aureus. So it's kind of a living, it's definitely an evolving field, because we're discovering newer ways for these bacteria to either evade us or help us, and it's a balancing act. We want to be able to have them help us more than hurt us, but that's where it comes into with the medical field, is discovering antibiotics to treat stuff too. So that's a pretty fun one.
Yeah, the idea of antibiotic resistance and gram-positive and gram-negative bacteria and other things. And we had mentioned this before, but like one of the beauties of STEM and science, more particularly what we're talking about, is just um, the different directions you can take. Like whether it's microbial work or you had talked about how you were initially like studying for the MCAT and you wanted to get into like becoming a physician in medicine, and then now you're switching a little bit here and getting into microbiology. It's just like there's so much. We talked about there might be not. You might think like, hey, how did we come up with this experiment and get to here? Like, or you have that like doubt, the self-doubt where you're like, oh, I can't do something like that,
Yeah.
But I think there's just so much to discover out there still that makes it very encouraging to enter into science.
Yeah, it's I think one of the greatest things that STEM allows you for, and we talked about before, but just variability, being able to do so many things with, even if you have just a single interest, you can apply it to so much stuff. So like for me, I love the environment. I'm a, kind of a, as people would say, granola. I like outdoors stuff, adventures, hikes and whatnot. And so one of the things that I love is the natural wonders of our national parks. And you see stuff like the United States Department of the Interior or a National Park Service, like they have these positions open where it's like, you think that the only thing that they would have present are like park rangers or people like park custodians, like ones that would like take care of the park and upkeep it. But you can look at, they have positions available for park microbiologists, going in there and studying geothermic reactions of bacteria living, subterraneanly, and how they affect, the environment, the ecosystem. Yellowstone, there's like sulfur pits there that, emit really high temperatures, extreme environments. And yet we finding sulfite reducing bacteria there that, contribute to heavy metal digestion in lakes and, removing pollutants. Like, there's so many avenues I just mentioned right there. Like, you talk about Yellowstone, and you talk about, getting rid of, dangerous pollutants in our water systems. Like, you took that from a single topic of microbiology, and you could say the same thing about medical field stuff too, as you find one application for a bacterial genomic project or something like that, and you can apply it to something that we know today as CRISPR, being able to identify cut genes. Like, the list is endless, and that's in and itself is an amazing thing. And yet I'm still not completely sold on it, but CRISPR is one of the most difficult to understand yet miracle based technology that I think I've ever come across as a scientist. I don't think I'd ever be able to physically do it because it's just it gets so convoluted in my brain, but it's incredible. The strides, leaps and bounds that we've been able to do because of CRISPR is nuts.
So let's take a step back here, because a lot of people might not know what CRISPR is, and it gets into our jargon and just from understanding it. But what is it, and why is it so influential?
So CRISPR is a technology that we really, dwell on as scientists, microbiologists specifically, because it kind of started with bacterial genomes, but we use it to edit genes. And so when we want to, one of the main places that we see this in is when we are using E. coli to mass produce things. Okay. Because now, granted, when you hear the word E. coli, everybody gets scared because they think of, I think it's I don't even want to look it up because I'm gonna get mad at myself. But it's not the E. coli that causes, devastating digestive tract issues. It's not that. We have strains that knock out the pathogenic, the virulent pieces of it. And so we kind of have been able to manipulate this prokaryotic cell in order to mass produce things of our interest. And so I'm not sure the correlation. I don't know if this is the exact example, but we use CRISPR in the same conversation as mass producing insulin. And, before, the production of insulin relied heavily on pancreatic digest material from pigs and cows that would come from slaughterhouses, in the 60s, 70s, when, not the onset of diabetic treatment would, started, but because of the scientific revolution in gene editing and modification, we've been able to take prokaryotic cells and kind of, edit genes within them, as well as genes of the cell itself, to sort of create kind of a just a pretty cool modification that benefits people and benefits our society. So
you got me curious. I just Googled it quick, but this Washington University in St. Louis scientists in April, I don't know if you saw this, but had developed a way to use CRISPR to edit a mutation in human-induced pluripotent stem cells and turn them into beta cells, and then we're able to transplant that into mice and then reverse pre-existing diabetes. So, biotechnology, microbial work, and really science in general. Can you tell where we stand on science? I don't know, but really strong proponents of the possibilities of it too. And even with my experience in clinical research and as however whatever experience I had learning from that job too, it's also taught me we talked about, this long process that is science. And we were working with phase 1 trials there, which is healthy individuals. We had a couple first in human studies, but it's just this really long process that tons of public data out there too. You can look it up on clinicaltrials.org, I believe. But CRISPR, really interesting. Really interesting.
It is.
Stuff.
It's super. It's again one of those things that you look at. You sit down as a scientist. You look at it. You're like, this shouldn't work. Right? You're like, it's like reading stereo instructions. You're just like, what? And then it comes out something beautiful that has just increased not only life expectancy of people on Earth, but just the scientific community. It's given a better understanding of how things work, and I feel like that's just one of its hidden gems of a kind of a benefit of understanding. And we kind of went down the rabbit hole of CRISPR, but
it changes.
It's a really good example.
It helps with understanding cancer research and things like that. So tying it into our partnership in May.
Yeah.
It's like it's basically a scientific scissors, right? That's able to like splice genes together. I mean, there's a whole nother ethical debate off of that, but
exactly. But again, it gets into the idea of the seeing is believing thing, and it's so hard for even myself. I've been able to witness sort of the CRISPR process in a couple of experiments, or in articles about this kind of experimentation style and technique. But it's still so hard for me to just comprehend, because again, we're looking at something, we're looking at DNA inside of something that we already can't see. And DNA is in itself is difficult to see. It's just like, it's a conundrum, a mental pretzel for me.
Yeah.
And so
shout out to Drew.
Yeah. It's just, it's hard for me to understand. But again, I do it for a living, and so not CRISPR specifically, but the stuff dealing with things that you can't see. So you you just have to take it out as. Now the funny thing is when I say that you can't see it, when I'm working with the cells, there's a way that you're able to see it, and it's how you grow them, and they grow in colonies. And so what I'm talking about really difficult to see, I'm talking about the single cell, a single bacterial cell. It's really hard to differentiate from a bunch of other things floating around in solution, because they're real. I think translucent is the correct word. Varying shapes sometimes, and again, they're living, so they can, move, float in solution. So but I've been able you can actually see bacteria when you grow it on a plate. They grow in colonies. So that's what I'm trying to get at. There's a difference. Like when you're looking at under a microscope, like, what is this? Well, when it's in, human tissue, when you take a human tissue sample and there's stuff floating around, it's hard to differentiate between other stuff, because again, they're they're microscopic. That's why, hence the term microbiology, because it's stuff very difficult to see, trying to understand the biology of it. So
yeah, now I'm deep into the rabbit hole. It's like
the
availability of it too. Like high school students now theoretically can alter a genome. Like
I can take an online course right now through Coursera about the genome sequencing, and like anybody could sign up for it and take it as a specialization. I would recommend having some sort of background in microbiology or biology or genetics or something to understand it. But like, you can literally whole genome sequence something from your computer right now. Like anybody that has a public or access to the internet can go and look and figure out how to sequence data, or like sequence genome data. It's nuts that one of these things that was probably inaccessible to people 25 years ago, unless you were a scientist specifically working on them, or you had communication with people that was working on it. You are now able to literally, I can type in whole genome sequencing of E. coli, whole genome sequencing of the human genome, or for example, too, the Human Genome Project in itself is a marvel of our scientific world. And again, you can definitely see where we sit on science, but we're like the science homers. Like we love this stuff so much. We're like, science homer. Yeah.
Science. Why don't we get into our little break here, and we can get back to what you're working on, and the ups and downs of that. So we'll catch you on the back half of this one.
Yep.
All right, so let's get into what you're working on, because word on the street is, looking to get published, not as like a first author or whatever, but so you're still on campus, or you're going up to campus to finish this independent study. Is it an independent study? Like, how would you classify this?
Yeah, I'd say that. I mean, I got credits for it as a class, and I had to present my findings at some sort of forum, and so it was probably as close to a definition of an independent study as you could get. They'd never really called it that. It was just research credits. So, what I mainly focused on was Columnaris disease in Salmonids. And so what that is in itself is a bacterial infection of, your favorite household name fish. You got walleye, river cat, some species of salmon, as well as some trout species too. So you have all these types of fish that are affected by this disease. And what it is, it's a bacteria that infects the fish via entry through the gills, or sometimes can digest the mucus on their scales, on their, and enter through that way and form an infection. But it actually looks like straw coming out of like so the term, I guess the general cell shape that the Flavobacterium is kind of given is straw-like, and so that's kind of where it comes from, is they look like straws, like pieces of straw coming out of the fish. It's in itself pretty disgusting and weird when the infection. But I'm not working specifically on infection. I'm actually looking at how the growth media of these bacteria can contain the proteins or the toxins or whatever viable for the infection to actually occur. Because what we find in a lot of infection with, like for example, humans, that it's not like the bacteria themselves are like, like they have teeth and like they're chomping down and biting us. A lot of the times, the things that are hurting us are enzymatic proteins, or some sort of digestive material, or some sort of endo or exotoxin that these bacteria can secrete that affect us. And so a lot of it is biochemical related. But what I've been doing is looking at protein concentrations in the media of these bacteria when they're grown, certain mutations of these bacteria as well, and seeing if there's stuff that isn't present that can cause disease, or if that's mainly the focus, is the stuff that's not there. If we knock something out, do the bacteria still kill? That's the premise of our experiment. And so when we find something that doesn't kill them when we knock it out, that's kind of how we know we got to look at this sort of protein complex and figure out the concentrations and stuff like that. So it's in itself pretty neat. I never thought that I'd get into it, because again, I was cancer biology all the way. I was pre-med. And so again, kind of tying some of my interests together. Like, again, I love fishing. Fortunately, I've never seen, a Columnaris disease infection in fish. So that was new for me, but I could understand the devastating effects, because for the longest time I was kind of environmentally aware of, aquatic ecosystems and the degradation of that, because I live in rural farmland, and you hear about the runoff of fertilizers and dangerous chemicals and how that affects ecosystems. So I kind of saw a little connection there. But this one was fun because it originated as an attempt to develop a vaccine against this bacteria. And so that's hopefully where my preliminary research and project kind of data is going, is for some sort of vaccination against the bacterium that causes this disease. And we can get into the subtypes and all that sort of stuff, but that's pretty in the weeds and pretty specific. It's just about as specific as the CRISPR mechanism that we just talked about. But yeah, I guess kind of just explaining it a little bit more, the process itself taught me a lot about learning from failure. And that's what a lot of science is. And we talked about that in last week's episode. But I would go in, this was when I was at school, I would go into the lab for probably 6 to 7 to 8 hours after my classes and sit down and run experiments, and conduct experimentation on finalizing a technique. Because if that was still up in the air too, this is a new strain of bacteria we're working with, so we don't know if the techniques established from previous strains still works. And we found out that sometimes it does, sometimes it doesn't. So it's really hit or miss. And it made me realize that you have to be pretty flexible with this. That's like a mini life lesson in itself, is like these things humbled me as a scientist, because again, you get the idea that they're invisible. You think that, well, they're a lot smaller than me, so I'm going to be able to manipulate them however I must. But almost always I was at the will of the bacteria. They had some dominion over me that I could not, conduct my experimentation and my technique and all that sort of stuff unless they said so. It was very manipulative on their end, if I'm going to be honest with you. So it taught me that, like, I can't take my, human sentient capacity for granted. Like, I have to be reasonable here. And so it was difficult, but the process itself kind of worked itself out. So yeah, but I still feel the challenges. Like, the other day an experiment completely went bad, But I also wasn't having a good day. Probably had like heat exhaustion or something. It was just overall not good.
Yeah. Oh, excuse me. Sorry, unrelated. I'm not yawning because that was boring. No, I'm really interested in a couple things. Like one, I want to go back a few seconds here because, right, like the traditional thought is that like vaccines are for viruses and antibiotics are for bacteria, right? But like that's not the case. Like there's for like whooping cough, diphtheria, and tetanus.
Probably one of the biggest ones that people get is the tetanus shot, and it's actually
the one you hear about the most too because it hurts, right? Like
Clostridium tetani. That's the bacteria that you're preventing because it's a soil-based organism, and so
I wanted to clear that up because you know, like even when you think like, are they actually vaccine? Like yes, like they're there's bacteria vaccines.
And want to know the funny thing is like the whole big conundrum about the anthrax vaccine. Anthrax is bacterial endotoxin. I'm almost positive is from Bacillus anthracis. Sorry, there you go. My professor would be proud that I'm riddling off these scientific names.
Extra credit for the degree.
Yeah, please help. Sitting right up there. Again, that's a really good point. There are bacterial and viral vaccines. Like it's not like one is associated with the other. Now, granted, antibiotics are only for bacteria.
Yeah, clear that
up. You hear that misconception too. It's like, oh, I have the cold. Let me go get antibiotics for it. No, the cold is seldom, if ever, caused by a bacterium. It's always caused by a rhinovirus or a coronavirus. That's one of the two common forms of the cold. And so you don't take antibiotics for that. If anything, you take an antiviral if you catch it right in time. Again, I'm not a doctor. I'm not trying to tell you, but like
antivirals are separate from antibiotics.
Yes, exactly.
And antibiotic resistant bacteria have an extra layer of
a whole bunch of stuff. That's
gram-negative, positive, like different staining in
that does. Sometimes like MRSA is a great example of antibiotic resistant bacteria because it became resistant to methicillin, which is the bacteria or the antibiotic that was most commonly used to treat staph infections. So that's a pretty big problem that we're coming across as microbiologists is antibiotic resistant bacteria, and it's important to know.
Here's a place where I can insert some of the things that I've been going through.
Exactly.
And we can kind of fulfill that promise that we made in the beginning of the episode. But I'm reading these accounts of pioneer doctors, and there's just this huge project, like I said, in 1955 about, in like May of 1955, they go all around Oklahoma and they start interviewing pioneer doctors that are, 70 up now, right? Like they come to Oklahoma right when, on the verge of statehood, they stay here and then they kind of see this modernization, quote unquote, it's 1900, but happen around them. And and they go from having horse and buggy to Model Ts with no roads, and they can't, there's no bridges, so they have to keep their horses in the, and the horse is basically swimming to get to the other side to get to this OB case, let's say, or whatever. But throughout like the whole thing is this like stigma against hospitals being like the end all be all. So like as soon as some of these small towns get like their first 12 bed hospital or whatever it was, there's the stigma against it that they in their words, it was like this prejudice against it, that they won't go to the hospital unless it's super dire, because they heard 3 people went to the hospital last month and they didn't come back. The same thing happens with these inoculations and immunizations, and diphtheria and other like whooping cough and things. I'm reading these public health reports, and the numbers are going way down. And by like 1909, there's like this push for vaccines and organized medicine to kind of take over and say like we have to vaccinate and eradicate these things, like they're all preventable. But like that there again, like it's a bacteria vaccine, like it's an inoculation, whatever you want to call it, immunization, whatever. But that here's where these germs, as they refer to them in germ theory, is kind of sustaining itself in public culture at this time. And there again, like they couldn't see it, so but what they could see and what they could fathom was like losing someone going to a hospital, not necessarily taking care of them if they're on their homestead, let's say, and they don't take their medicine or they don't keep their rooms well ventilated or whatever it is.
Yeah, it's whatever. Yeah.
But here again, like it's something that, you know, I don't want to use repeats itself, but it comes up again in our, you know, historical timeline, and you know, it's all these microbials' fault.
Yeah, these darn things.
I know,
And
but
then,
they do so much for us too. So it's like you gotta
and that's
symbiosis.
Another up and coming area of study that was mentioned during my time as an undergraduate and kind of in high school, but the idea of probiotics, you know, the bacteria that are actually fighting for us.
Mm-hmm.
And, you know, you hear, I was mentioning before, the skin and the digestive tract, like the amount of things that we still don't know about it compared to what we think we know is remarkable. And, you know, like you hear, well, I drink like a probiotic yogurt booster thing just because it's, you know, I know kind of the benefits of it, but you still hear the idea of like, oh, I'm ingesting bacteria. It even goes as far as the labeling on these sort of probiotics, because they'll have lactobacillus on there. They'll have these thermophilus bacteria on there. But one of the ones that they have that is kind of terrifying is a strep bacteria, but they won't even put strep on there, because when you hear the word strep, everybody's like, pandemonium sets in. They're like, oh my God, I'm gonna get strep throat from eating yogurt. And I've gone in the weeds of this and understood, like, it is almost, I don't like saying the word impossible, because I've found a lot of things that I thought were impossible actually happen with bacteria, but it is next to impossible that the strep in your yogurt is going to either mutate to a virulent strain that gives you strep throat, or is itself strep throat. So but it goes to show you like how powerful these bacteria are. Is like it even affects the labeling on a household dairy product.
Mm-hmm.
Like that's nuts to me, that like these like that it stretches that far to marketing, like bacteria and marketing. Like, whoa, what? But you have to be careful because people will. I'm not saying, because it's not like I've never freaked out. Before I knew about it, I was like, hey, wait a minute, like I would have been scared of like strep throat, right? But it's like a misconception that because these things are labeled this way, they mean this. But you know,
did you get strep a lot when you were a kid?
The first time I got strep was this last year, and that was before you know, the whole thing.
I was one case away from losing my tonsils.
Tonsils were Audi 500. Yeah. That's a fun topic.
So the other thing, remember I had two things, right?
Yeah, you did have
the other thing.
Two.
One of the things. Okay, the second thing is your research, right? When you were talking about your research being maybe a chance for a vaccine or like a treatment for this condition for fish, right?
Yeah.
You yourself are not going to be the one, maybe, who knows, but you
who knows,
probably by the time you get your degree and like run your own lab and like maybe this will already be done, like whatever. But the larger point that I'm trying to articulate is you are adding to this greater community of science.
Yes.
Right? Like you yourself now are a part of this you're a part of a couple of different things, right? You're a OU student. You are this part of this like graduate science student community. You can join, you know, other I don't know what kind of society, like
ASM is one of the biggest.
Okay, so like
American Society for Microbiology, it's huge. Yeah, it's huge.
Yeah, so like you can, not saying you could be a member in undergrad, but you can do that now. Like there's you're putting yourself in this larger community and situating your research as kind of this like throwing like a let's use this example, like skipping a rock into the ocean, right? Where you're throwing in your experiment and your paper that you're, you know, going to be published on hopefully, and you're adding to this like greater pool of research. And I think that is like a super cool like way to think about it too. Like even with like my stuff too, you know, hopefully I can find somewhere to get published for my thesis when I write it next spring, and you know, throw my hat in the ring of this larger community, this academic community, this scientific community, whatever you want it to be.
Yeah.
And you have a connection to it that, you know, nobody can really take away from you unless they kind of really find big flaws in your history or your science and in your methods. But I think that is like a super cool way of conceiving, not just being, you know, a student or in academia, but just like as a whole, it's cool to be a part of that.
Yeah, and it's great. It's like it's a network again of people you can rely on in the sciences that have accreditation, that have some sort of truth in what they say about a topic. Because when you're in the in the thick of research, right, like when you're going in there pretty much into the unknown frontier of whatever you're trying to get into, it feels better to know that you have people behind you, next to you, in front of you, doing the exact same stuff. Now granted, the experiments are different, but they're doing unknown science for the first time ever in the history of humanity, and you're able to go in it with this network of people to collaborate with, because that's really the beauty of science.
That's a really great way to talk about that.
Like, it's essential. Collaboration is essential. No matter how much I wanted to say my research was independent, because granted, I was in there doing the man hours. If I didn't have my advising professor, if I didn't have the people that wrote this journal about glide and motility, if I didn't have producers at VWR create me like my flasks to do this stuff, like, you don't realize how much of an essential network you rely on, as like, unless you're actually in it. Unless you're in the in group, right? And I'm not meaning in group as some sort of social class, but but it's like you don't realize that, because like before I wanted to become a true scientist and like a microbiologist, like before I figured out that's what I want to do, I looked at science as, oh, this is really cool stuff. You're going to discover all these cool things. And I never asked well, how do you get there? Who's behind you? Who's with you? What do you need? Like, and then the quintessential question, why are we doing this? Like, that's the fun one in itself, is like, answering those why questions probably gives you more gratification than a lot of the things that I thought would in my life, is being able to answer like, hey, so why does this mechanism do what it does? Or, hey, I sat down and did this for a year and a half. I could probably answer that question. Like, being able to answer those whys is a pretty powerful thing. And I think that's what, with the help of a graduate society, for example, or an undergraduate society, or any of these, cumulative groups for learning, what they can help you accomplish is outstanding, because again, you can't do it alone.
Absolutely. And I really liked how you put it, because it is this like larger community, and you don't think about that when you are first getting into it. Like, you don't think about who you're going to need to kind of like support you, and whose work you're going to build off of, and who's going to, review your papers and edit it for you before you submit it to a journal, let's say, and like you said, like, knowing that, let's say, traditionally your project should be a little bit different than someone else's, right? And knowing that, and finding ways to, you represent it as this like Venn diagram, where you're like, your thing is right in the middle. No matter how many circles are outside of it, you can, collaborate
you're pulling from so many. Yeah,
exactly. At the end of the day, it comes down to that center circle, which is going to be your stuff. But, knowing and understanding people like that are interested in the same things that I do, reaching out and just like saying like, hey, I really appreciated how you did this. Like, do you have any tips as I go forward in my research? And like, you'd be really surprised how many people would be so willing to just say, hey, like, best of luck. Like, here's what I did, and I think that's a really good place to close this one out, is understanding, this network around you that helps lift you up.
Yeah, exactly.
And get to a point that you can kind of, not that you overcome imposter syndrome, but like, you get to a point where you're like, hey, like, I spent a year and a half working on this. I found this, and this, like, I have things to rely on, that you don't give this like super political answer. You give this like well thought out hard work that goes into this answer. And like, when I'm asking you about your research, and like, you can hear about like, so this person did this, and then we had to do this, and it was really long, and painstaking is my new word for the past couple of weeks, but it's just like this long process. And I think it's really, like you said, it's a really nice way to kind of articulate.
Yeah.
Covey.
You can hear Covey in the back. That's perfect.
He said, wrap it up.
Yeah. And just real quick, one of my most favorite things about it, and it's the less talked about part of science, is the science that goes wrong. The stuff that doesn't work. And for the longest time, I don't know if there's still a publication for this, but there was actually a publication for scientific methods and experimentation techniques that don't work. Mm-hmm. And people actually submitted their real life results that they got and submitted it to this journal, and it was published. And scientists like myself, new to the experimentation process, well, in the lab setting, right? I was able to like think about an experiment using FIAC, right? Like that's what it is. Or problem, hypothesis. Oh my God, observation, experiment, conclusion, FIAC. Yeah, there it is.
Sure, man.
Yeah, whatever. Anyway, just a big shout out to that stuff, because there were certain methods that I would have spent hours of my life trying that wouldn't have worked, because somebody before me said, hey, I spent 2 years doing this research and I found that this method works, this one doesn't. And they'd publish that for me. And it's like, that just goes back to that network of like people not wanting other people to fail, because it's the most essential, but at the same time, it's the most degrading part of science, because it just makes you feel like you didn't accomplish anything, but in reality, you know what not to do for the next time. And that's almost as powerful as, a new discovery. But
For sure.
you can't, I want to say this firmly, is I don't think you can do science alone. And I think the collaborative effort that we have in these, I don't want to say graduate society for learning and like make it all, but it really does feel like its own society, like its own community of people that are just out there to interact, help communicate, and learn with you. And it's nothing more can be said about it, because it's just that beneficial to me and to us and to scientists everywhere. So it's a really great thing.
Absolutely.
Yeah.
All right. Well, we'll close it there for today. We thank you guys. We'll catch you next week.
Yeah. Peace.
Well, if you made it this far, first off, thanks. Second, we hope you enjoyed this conversation. We're always looking to grow our content. We'd love to hear from you. Be sure to, like, subscribe if it's on YouTube, or, follow us on, I think they do follows on Spotify now for podcasts. And, we'd love to get your feedback and hope you enjoyed and learned something from this conversation.
Yeah, it's always been a blast talking. That's why we do it, and that's why we enjoy doing it. And it's always fun to talk about stuff that we're really passionate about. And so I'm hoping that if anybody ever has any questions, comments, concerns, that they can talk to us, because of, like I said in the beginning of this episode, we're experts, but we're not close to the experts that we feel confident in standing completely, firm on what we say. Like, there's some things that, yes, we do believe in to be true and, undyingly true, but there are things that we're wishy-washy still on. And so if you guys ever have anything, just always interact with us, because we'll appreciate it. So
Bye.